Hello, everyone. So Anne-Marie, can you confirm you're seeing my full screen?
I am seeing your full screen and I'm hearing you loud and clear.
Perfect, thank you. All right, well, everyone, welcome and hello. My name is Natalie Steinauer and I'm the research coordinator for the Be-Informed Partnership. And today with some of my colleagues, we are presenting on adult bee disease and how to diagnose them. This is the second part of a two-part mini-series on bee diseases. So following the webinar that we recorded a few weeks ago on brood diseases, and then another one that we did on varroa specifically. So if you missed our webinar, you can find those recordings on our website as well as on our YouTube page. One of my colleagues will put those links for you in the chat. Okay. So thank you again for joining us today to learn about how to diagnose adult bee diseases. We are the Be-Informed Partnership. Our mission is to improve colony health, and we do this by bridging the gap between scientists and industry and supporting beekeepers in their practice. So our field specialists presenting with me today work specifically in our tech transfer team program, which is one of our many programs, but mostly focused on supporting large-scale operation and commercial beekeepers in all of the big commercial beekeeping hubs throughout the country. So working alongside commercial beekeepers, they have had their eyes and hands in hundreds of thousands of hives, of colonies, and they have accumulated years of experience, and we want to share some of that experience with you today. So in this webinar, we will address the following topics: CCD, basically what it is and what it isn't, but also honeybee viruses, Nosema, and tracheal mites. And each of those topics will be covered by myself and then some of our tech transfer team field specialists, Rob, Kate, and Dan, respectively working with us from California, Texas, and Michigan. So we wanted to start off by really addressing the elephant in the room and rectifying a lot of misunderstandings surrounding the issue of CCD. Now, if you search CCD on the web, you cannot not find press or published works that alarmingly describes the doom that awaits us if all of the honeybees were to disappear. And you know, now we all agree that there are concerns surrounding honeybee health and just environmental quality and loss of biodiversity in general. But it is important to really separate the issues and define the issues so that we know what we are talking about. So I would like to start with a poll to kind of like judge where you stand on the following statement. Which of the following statement is correct? So we'll give you a couple of seconds to address which of those statements you think is correct. And then we will address that and then tell you which one is correct and then why the others are wrong. All right, we're reaching 80% soon. All right, so thank you for answering this. We can see that the majority of you have answered correctly, that the number of colonies in the US is stable despite high mortality rates. A few people among you are worried that the honeybees in the world is decreasing, that honeybees are in danger of going extinct, and that CCD is the major cause of colony mortality. Those three statements are wrong, and we will cover all of those in this presentation. And now there is a second poll that we wanted to ask you very shortly, and it's, have you ever observed colony collapse disorder in your own colonies? All right, we're reaching 80% really fast, and the majority of you have not seen CCD in your own colony, while some of you, 18%, have observed CCD in your own colony. So we will actually address what we consider is CCD, and then we will ask you again to see if you then can confirm that choice. So we obviously are all in agreement that honeybee health is not ideal. There are issues, there are concerns, and we at PIP, we are documenting the turnover rates of colonies every year. We have run the loss and management survey since 2007. A lot of you have mentioned that you have answered the survey this year, and we thank you. So we know that every year about 4 out of 10 colonies in the US will die off and need to be replaced. So this shows a mortality rate, right, which can be matched by something like a natality rate, right? So for colonies, that means that creating new colonies by having swarms or making splits can be the counterpart of losing colonies. So it is not the same as a population loss. It is really a turnover. A lot of people are worried that honeybee populations are in decline. And to look at that question, we need to estimate the total number of colonies in the country. And this is something that the USDA does every year in their honey reports. So looking at those numbers, we see that there was a reduction since the early 1960s, in which the US had about 5.5 million colonies. And more recently, we have been hovering around 2.5 million colonies in the United States. Now, we can talk about this graph in more details, and particularly about how the incidence of CCD and the arrival of varroa mites in the country, they have barely impacted it. And by comparison, the biggest drop in population estimate was actually due to a methodological change in how the survey was conducted. But in short, I have added here the arrow showing since when BIP has conducted his loss and management survey. And so since BIP survey started documenting 40% annual turnover, the population, the total number of colonies in the country has remained stable despite high mortality, despite high turnaround. So that means that beekeepers can maintain their operation size, they can recover from their losses, and the population as a whole is relatively stable. Of course, that comes at a cost, and we don't want to underappreciate the efforts it takes beekeeper to do that. So I just wanted to give you in comparison, to give you context during the same time period. So this is, in orange, we show exactly the same data as before. The scale is different because now I'm also showing you the European Union and the world numbers. So this is the estimate of the world number of honeybee colonies. And you can see that honeybees are actually increasing in the population worldwide. So there might be some regional declines, but overall honeybees are neither in decline nor at risk of extinction. Again, this is despite high turnover on the colonies, because beekeepers can make new colonies and maintain or increase their operation size. So this is the first, I guess, main point from this presentation, is that whenever you talk about colony loss mortality rate, it is different than honeybee population decline, and it is different from CCD. So those are 3 different phenomena that have different drivers and different consequences. So what about CCD? Well, okay, CCD was an outbreak. It was a cluster in time and space of high mortalities with really, really weird features. The first cases were declared in the fall of 2006, and then it became more widespread in the spring of 2007. And the beekeepers were reporting large and unusual die-offs. Now, what was unusual was the way those colonies died. They described that thriving populous colonies would be found mostly empty in a few weeks, with no dead bees that were found in or around the hive. Now, that drew a lot of attention because this is why we started hearing about the vanishing bees, right? Those bees just suddenly disappeared. But actually this particular sign, no dead bees found, was actually not completely unheard of. It is actually called altruistic suicide, and the bees that are sick are known to do this. So they will walk away, they will die outside of the colony. And this has been observed in multiple infectious cases. For example, high varroa infestation can trigger altruistic suicide of adult bees, and other infection can cause that behavior as well. So it is believed to be an evolved response to minimize infection inside the superorganism that is the honeybees. So that on its own was not unheard of, but what was really particular, and so when the researchers described CCD, they actually coined several key signs that all have to be present in order to qualify as CCD. So all the colonies that found dying or dead from CCD, they shared this common set of clinical signs. The first one was the rapid loss of adult bees from the colony, as indicated by large areas of brood that remained unattended. So you can see in the figure above, we basically had excess brood population relative to the adult bee population that wasn't enough to cover all the brood. And this was really rapid. As I said, in a couple of weeks time, you have this rapid loss of adult bees. The second, of course, was the absence of dead bees in the colony apiary, as I said, probably due to altruistic behavior. And then the other sign was the fact that there was sufficient stores of honey and pollen in the affected colonies, meaning that they didn't lack in food, they didn't starve. And again, another sign was the apparent delay in kleptoparasitism in abandoned hives. So that means robbing or invasion by small hive beetles or wax moths. So you probably all know, if you keep bees, that if you have unattended frames inside a colony that doesn't have bees, they will get attacked by those opportunistic pests, and that can arrive very, very quickly. So that was a weird symptoms in which that kleptoparasitism, those opportunistic pests, would either not come or come really later. So despite the fact that basically the food stores were undefended. And then finally, a little later, they added to the definition that the levels of Nosema and Varroa had to be insufficiently high to explain the collapse. So they ruled out Nosema and Varroa from CCD cases. They ruled them out as causes, and they said, in order to qualify as CCD, you have to have low infestation of those two pests. So you can see how each of those signs can actually be expressed in multiple bee health conditions, but all together they describe CCD. So having one or two is not enough. You really need to have all of them to qualify to the strict definition of CCD. And so then you can see now how this term can be misappropriated by a lot of the press out there, and what the issue is. So we talked about that the first case description was done in 2007 in the Cox-Foster publication, which I've shown a print screen here. And then researchers actually looked at historical reports, and they found that such losses actually had been documented in the US at least 18 times in the last 110 years. And they were reported under different names like May disease, disappearing disease, and never really clearly defined. So when it was defined in 2007, that's when they made the case definition, listed all of the signs, and made the actual case paper. So to date, it actually has only been documented with verified case only in the US and in Switzerland. And still today, no definitive cause of CCD has been identified. The original study implicated a virus, which was IAPV, in combination with Nosema as a potential causal agent. But then the association was not confirmed by a second, larger study that had more samples, and that actually followed over 200 different variables to try to find a source that would explain the cases. And still no single factor was found in all cases. They even found that pesticides that are often suggested as a putative cause for CCD were higher in healthy colonies than in colonies affected by CCD. So still a lot of unknowns. They did notice that colonies dying from the condition were clustered together, so hinting that the condition seemed communicable, or the result of some common exposure rather than some large-scale change in the environment. And they also found that the bees collected from collapsing colonies had higher pathogen loads and were more often co-infected by multiple pathogens rather compared to healthy colonies. So it did suggest that maybe the immune system of the bees were compromised, letting then the bees succumb from opportunistic second infection. So a little like AIDS in humans. So what now? Most experts, most apiary inspectors in our country, and our own field specialists at the Bee Informed Partnership agree that we have not encountered cases that meet the strict case definition, at least in the last 5 years, if not 10 years. Our oldest BIP field specialist team was established in 2010 in California, and none of us have ever seen an official case of CCD. So definitely CCD is not that prominent anymore. It really acted like an epidemic. It had a peak of case in 2006, 2007, and then stabilized back under detectable levels. We are still unsure what the cause is, but we know that it is not a major cause of honeybee mortality anymore. However, it did act as highlighting and attracting the attention of the press and the public to long-term issues on honeybee health. So it really acted like a whistleblower. But it is, unfortunately, has been misused in the press to mean more than it actually is. So if you really follow the case definition, CCD is not around anymore. It was a flared epidemic. It is in line with a virulent pathogen hypothesis, though the original causal agent has not been identified. It is possible that it might flare back up from time to time. And if it does, we hope that then the new molecular analysis will allow us to identify a causal agent this time around. But currently, it is not something that we see. Now, unfortunately, if you use the definition as used in the press, which is mostly misappropriated as any problem touching honeybees, any lost colony, then yes, I guess CCD continues because we do see high loss rate in many places. The problem of using the term in that way is that it kind of hides the fact that with a proper management, many colony loss can be averted. So it is important to disambiguate issues because there are some issues that can be averted, and hiding them under catch-all terms kind of hide that fact. Now, when we ask beekeepers, however, a lot of beekeepers still report CCD as one of the cause of colony loss in their operation. I'm showing you here over 8 years of survey data in which we ask the question, what do you perceive as the major cause of colony loss in your operation? And we noticed that about 29% of large-scale beekeepers have reported it. It is actually the fourth most important cause of colony loss reported by large-scale beekeeper after varroa, queen failure, and pesticides. In comparison, only just 7% of small-scale beekeepers report it. But so we can see that it is a perception problem and probably is a definition problem. And that's why people are still reporting it. So we know that colonies can be lost by, due to a lot of different factors, and a lot of things can go wrong in a colony, but it is important to try to identify the reasons a colony die, because that's how you can try to make sure that it doesn't happen in the future. That's how you can understand the issues and remedy it in the future. So over the years, our field specialists have been called to sites where people have reported CCD, but they always found that there were other stressors, other causes to those issues. So remember the list of signs associated with the definition of CCD, and remember that all must be true to qualify. So if you do have dead bees in a bunch at the entrance of the colony, then it is not CCD. If you have high mite infestation, it is not CCD. If you find an empty colony, but it doesn't have any brood left, it is not CCD. So finding a dead colony in your yard, we know it's a harrowing experience, and figuring out what killed it is tricky. It does take experience. But we hope that it's an exercise worth practicing, and we hope that we can be helpful. For example, in this webinar, where we can try to give you tips on how to do this identification. So I'm going to finish my part by asking you the same question again, which is, have you ever observed CCD as defined here in your own colonies? All right, thank you very much. So we have the majority of you that are still reporting no, and then we will check to see. We have less people that are now reporting yes, but again, it might be also all cases, we don't know. But I thank you, and I'll pass it on to
Hi, my name is Rob Snyder. I work on the Northern California Tech Transfer Team, and I'm going to talk a little bit about viruses and the way they're transmitted and some of the visual symptoms of some of the viruses that we see in the field. Go to that next slide. So since the introduction of varroa mites in 1987, viruses have become more virulent, and beekeepers pay much more attention to their colony's health, the varroa mite treatments, and the management of these mites. Once viral loads are high in colonies, it could take 2 years or more to rid the colonies of high viral loads through diligent varroa mite treatments, requeening, and supplying bees with good forage or food substitutes if they're in poor forage areas. And the best way to keep viral loads at a minimum is to monitor varroa mite levels and treat colonies when they're close to a threshold of 3%, although in the spring you want your mite loads to be around 1% or less. So here I'll talk a little bit about an overview of the transmission, and then I'll get in depth in each one of these sections a little bit. So you have horizontal transmission, which is basically from B to B or contact. And then you have vector mediated, which means that it's spread through varroa as the vector of the virus. And then vertical transmission, which would be from mother to daughter or father from the queen. So viruses can be transmitted through different routes. Traditionally, viruses were transmitted horizontally from B to B, either through oral or fecal route or simple contact. But viruses can also be transmitted vertically from mothers to daughters. The transmission route is an important factor that determines the dynamics for these pathogens. Viruses can also transmit in indirect ways using vectors like varroa mites. The recent spread of varroa destructor greatly modified the epidemiology of several viruses, which switched from mostly orally transmitted pathogens to rapidly spreading epidemics driven by the direct transmission or indirect effects of varroa mites on individual colony bee health. So shortly after this invasion of a virus such as ABPV, DWV, or SBPV were shown to be successfully transmitted by the mite, some of these viruses rapidly evolved towards vector-borne lifestyle, which increased the rates of prevalence and virulence. And here in this slide, you can kind of see the different ways that the viruses are transmitted. You know, the top one is showing, you know, from larva to adult bee, and then you could have cannibalism and feces being a way it's transmitted. In the next few slides, I'll just show some more here of the transmission. So on this next slide, you could see, no, I think you skipped one ahead on me there. On this slide, you could see, you know, multiple transmission routes, and you'll see from the egg to the queen or to the drone, those are vertically transmitted viruses, and that's one way that the viruses can be transmitted is from the queen and not necessarily from varroa. But onto the next slide here, varroa mediated transmission from an experiment that Kelly did. Basically, we found that colonies that had higher varroa mite levels on average had higher virus levels in the fall. And here is just showing the fall and the higher viral levels here in the orange.
Rob, your microphone is breaking up a little bit. I don't know if you're in an area where you can be closer to your modem or something, maybe?
Yeah, sorry. Is that better? Okay, so this next graph here, on the left-hand side, we're just showing you the viruses that we test for, and the graph on the right represents what a viral report would look like. And this is IAPV, or Israeli acute paralysis virus. And from this sample, if you look on the far right, there's a line that says that you're in the 94th percentile out of 100. So that means that the viral level is fairly high compared to the 1,057 samples that were taken to get this percentile. So these viral samples are relatively new, and like I said, there was only 1,057 samples that were taken for this virus. So it's fairly new, and we're still learning a lot about these viruses, and there is only a handful that have visual symptoms, and I'm going to talk about those next year. So the most common visual virus symptom is deformed wing virus, and here you could see the bees that are circled in red or pink or purple, whatever color you see there. You'll see those bees with the deformed wing, and then also you'll see in the picture on the left there's some of the brood that circled, and that was brood that had either died from a virus, or there might have been a mite in the cell, and the bees started opening that and cannibalizing it. And that's goes back to, you know, a way that these viruses can be transmitted is through cannibalization. And then you'll also see the picture on the left where the queen is there, and she's another mode of transport for these viruses. And if we go to the next slide, I'll show you some more images of deformed wing virus here. So here's just more symptoms of deformed wing virus, and usually a colony that shows these symptoms has mite levels above 10%, which is about 3 times the threshold that we consider reasonable. And if you see these symptoms, you know, the colonies should be treated right away, but often it can be too late if the virus levels are high, because once you have high viral loads, you can't really do anything for it. So your best option is to just monitor Varroa and treat for the mite. So here's a very common virus that shows up, and that's black queen cell virus. But often the only people that see this virus, like these images here, are people that produce queens, and they find this stage when they're candling the queens, when they hold the queens up to the light to see if the queens are alive before they decide they're going to put them into a mating nuke. And when they hold them up to the light, these ones are obviously dark, and the queens look as if they're almost filled with oil, and it's kind of similar to sac brood when you pull these out. And I'll talk about that virus in a few slides here. So another, I would call it an uncommon virus here, is chronic bee paralysis virus. And it's most noted by shiny bees, which usually there's about 25% or more of the bees on the frames will be displaying this shiny characteristic. And some may act normal, some may be twitching, some you know, may swarm out of the colony and be hanging in the trees, I've seen. And it kind of resembles a pesticide spray. And these are not to be confused with robber bees, which are also shiny, but usually there won't be quite as many of those in the colony, and you'll usually see robber bees in the fall. So, you know, this is not really common, and I've seen it about 4 or 5 times in the past 10 years. So I just haven't seen this much, and the cases that I've seen it, the bees were pretty isolated, and we were thinking that it was possibly transmitted by the queens, because there was no other beekeepers around the area to have given the colonies this virus. So that was just a theory that we had. So sac brood virus is probably one of the older viruses that we've seen symptoms for a long time. When I started inspecting bees about 12, 13 years ago in Pennsylvania, I saw quite a bit of this virus and the symptoms. And since I've moved to Northern California, I haven't really seen this virus much. And I think it's due to the hygienic stock that these guys have, and they're cleaning up these infected larvae fairly quick. But the characteristics are, you know, it's got this pointy head, and basically these are uncapped shortly after they are capped. And in the center picture, you can see there's one of these sac brood virus larva there with a mite right next to it. And then the picture on the right is a little bit darker as that one has aged. But when you go to the next slide, you'll see some of what this sac brood virus looks like when you extract the larva or the pupa out of the cell. It kind of has this sac-like viscosity where the liquid is contained inside this sac, where if you were to pull out a regular pupa, you know, it often break and just lice and the contents would just spill all over. Where with sac brood, you get this, you know, this defined little sac-filled fluid. It's sort of like a mummy, but it's not. But if you do pull out a larva and it is chalky, you know, that's chalk brood, which is another disease. But sac brood virus and chalk brood, some of the symptoms can look very similar. But these pictures were from Dan, who's going to talk in a little bit here. And next I'll talk about just how we collect these virus samples. On the left here in the picture is some of these viral tubes, and then on the right-hand picture is probably about 30 or 40 of these viral samples that we take from the brood nest, and we put them on dry ice right away so that the RNA doesn't break down, because the RNA breaks down fairly quick, and that's what they need to do the diagnostics on for the viral test, which is on the next slide here, where you switch there. So here's some of the samples being ready to be processed for the virus diagnostics. They'll separate the bees out of the tubes, and then they'll basically run those RNAs against some of the baselines that they have, and they'll give us a viral report, which was similar to that graph that I showed you on Israeli acute paralysis virus. And then we report those to the beekeepers, and there ain't much they can do when they have the viruses, so their best option is to you know, just maintain low varroa mites. And that goes to my last slide here, which is virus prevention and treatment. Unfortunately, if you have high virus loads already, there isn't much you could do besides requeen and try to get the colonies on good forage. So your best option for prevention is to monitor your varroa mite levels and not let the mite levels get above threshold. And the best way to do that is, like I said, is to just monitor month by month. And if you get above that threshold of 3, you're going to want to treat those colonies, and that should keep your viral loads at a minimum. And hopefully you won't see any signs of any of those viral symptoms that I showed, like chronic bee paralysis, sac brood virus, or DWV. And then I think we have a poll here. And have you seen deformed wing virus in your colonies before? Okay, so it looks like 53% said yes, and 37% said no, and 10% didn't have V's. So quite a few of you have seen this this virus before, and hopefully you're not seeing it now, and it was something in the past, and you've learned from it, and you've been maintaining your varroa mite levels. So I believe our next talker here will be Cade, and he's going to talk about Nosema.
Hello everyone, my name is Cade Houston. I'm the Texas Tech Team Field Specialist, and I'm going to talk a little bit about Nosema today. So before we get into talking about how Nosema is in honeybees, we need to know what it is on a biological level. So Nosema is a Microsporidia. Microsporidia are eukaryotic unicellular organisms allied with fungi, but they're actually placed in a sister kingdom simply because they're hard to place in the nomenclature with fungi. So they're intracellular obligate parasites as well. So intracellular obligate parasites affect the gut of the host. So obligate parasites cannot complete their life cycle without a host, as is the case with Nosema and honeybees. So the picture in the middle is a comparison of two different bee guts that have been pulled out. The top one you can see is clearly lighter in color than the one on the bottom, and that light color suggests that it is infected. And the picture on the right is a microscope look at Nosema, and that long yarn-like thing you see there is actually the polar tube, the polar filament that they use to infect the midgut cell. And we'll talk more about that in the next slide. Okay, so now we're going to get into the life cycle a little bit. So Nosema spores enter the midgut through contact with other bees via trophallaxis, which is mouth-to-mouth contact, or presence of feces in the hive environment. So once the Nosema cell reaches the folds of the midgut, that long polar filament, it'll shoot that into an epithelial cell, into a midgut cell. And it releases the reproductive organism there, and those cells begin to replicate, and they replicate until that cell is just so full it breaks. And those Nosema cells are released, and they go and infect other bees inside the hive. Okay, so the two types of Nosema that affect honeybees are Nosema apis and Nosema ceranae. Nosema apis was discovered on Apis mellifera, the European honeybee. It made its way into the U.S. by the early 1900s and was believed to be the only type of Nosema that affected honeybees for a long time. That was until Nosema ceranae came along. And the first report was in Europe in 2006 of Nosema ceranae. But it was believed to have been in the U.S. since the 1990s. It was first discovered on the Asian honeybee, and but now it affects other Apis species and also non-Apis species. So pictured here are on the left are Nosema apis and Nosema ceranae. So as you can tell, there's no way to distinguish the two species under a microscope. So molecular analysis is the only way to distinguish the two, and we no longer speciate between the two because Nosema apis has not been seen in years. Therefore, from now on, in this presentation, when we describe Nosema, we will be talking about Nosema ceranae. Okay, so dysentery was often associated with Nosema apis, but not with Nosema ceranae. Therefore, nowadays, if you see dysentery, it could just be a sign of spring cleansing flights if the bees have been stuck inside the hive for a long period of time due to cold weather or other environmental factors. Also, different types of forages will cause dysentery. So, if you see this dysentery, it's most likely a sign of another environmental stressor. Okay, here's some more potential signs of Nosema. Visual detection is very unreliable, but some of these signs include K-wing, disjointed abdomens, absence of stinging reflex, or bees crawling on the ground. Just do note that these are potential signs of other conditions as well. As Rob pointed out earlier, and Dan will point out later, other conditions such as bee paralysis, virus, starvation, and pesticide poisoning, bees do exhibit those signs with those other conditions. Okay, so here, this graph here is the Apis longitudinal data in regards to Nosema. So as you can see, in 2018, 65% of apiaries tested positive, or approximately 65% of apiaries tested positive for Nosema. This was assumed Nosema ceranae. So the infection intensity or average Nosema load, as you can see there in the graph, is measured in millions of spores per bee. And you can see that it fluctuates throughout the season. There is a slight increase in spring, which may be due to the bees are coming out of cold weather. They haven't had the chance to really get out and do their cleansing flights. So the levels might be higher. And also, Nosema is an old bee disease. So your older age bees inside the hive are likely to have higher spore counts. So you may have, your queen's just getting started, really just getting laying, and your brood production is really just starting back up. So the population in that hive is going to be a lot of old bees. So you may have, that may be a reason that we're seeing higher counts coming out of winter. Okay, so here, this is the graph that Natalie used earlier. And as you can see, on average, about 15% of large-scale beekeepers identify Nosema as a source of colony loss, and only 4% of small-scale beekeepers identify it as a cause of colony loss. And just another note, the fraction of beekeepers reporting Nosema as a cause of colony loss has decreased over the years. Nosema ranks 9 out of 16 in the order of most important causes of colony loss in large-scale operations, and 10 out of 16 for small-scale. Okay, so we have a poll here. So, do you monitor for Nosema? Okay, looks like about 66% do not monitor for Nosema, 23% yes, and 11% said, I do not have bees. Next slide, please. All right, so now we'll go into some Nosema diagnostic techniques. So microscopic examination is the most reliable and most used to account for presence and quantity of Nosema spores. So in our lab, we don't use dissection. So we take a sample of bees, crush them with a rolling pin, which makes sort of the insides of the bees are crushed out, so it makes sort of a bee juice. And a sample is taken from this, and it's analyzed under the microscope to quantify the amount of spores that are present. And you can do this at home. Just get a certain number of bees, crush them up, and take a sample and look at it under a microscope if you have one available. So those are microscopic, just to tie back, microscopic examination is really the most reliable and most used way to diagnose Nosema. All right, so now we'll go into some prevention methods. So to prevent Nosema, beekeepers want to have good brood production, and you can't have good brood production without proper nutrition. So you want to make sure your bees have enough honey and pollen. You want to ask yourself that question: do my bees have enough honey and enough pollen stores to successfully rear brood? You want to make sure that they have everything they need to make that hive grow, to make that population grow, and to really amp that queen up and get her to lay more. And so, I mean, you can feed through feeding pollen supplements, sugar water, corn syrup. You just need to make sure that those bees have enough feed, and if not, the management practice would be to feed them. All right. The next thing is you want to have a young, prolific queen, and that equals good brood production. So just that's another thing with the good brood production. You want to have having her in that hive will give you that large population of younger bees. As we described earlier, the Nosema is an old bee disease. You want a high population of those young bees to prevent the nosema. Um, and actually, in fact, high nosema spore counts oftentimes do go back to queen issues in many cases. So a good management practice would be to monitor the age of your queens and the production rates as well. Make sure those queens are producing to the best of their ability, and yeah, your hives will be that much better for it in nosema prevention. Okay, so next thing is choose good apiary sites. So you want to choose good sites with good forage, which goes back to good nutrition, which in turn goes back to the good brood production. So sites with good forage, proper ventilation, good water drainage, and good site accessibility. So like we said earlier, like we just said, the good nutrition goes back to good brood production. Nosema is closely related to fungi. So they like those moist, humid conditions. So good water drainage and proper ventilation is completely necessary to prevent nosema. And good site accessibility, that's more on a management for the beekeeper scale, because if you don't have good site accessibility, you can't get in there to feed, to medicate, to requeen if needed, and all these things play together in nosema prevention and are connected. So it's good to have these three. Okay, so now we'll go into some ways to treat nosema. So there are two ways. You can eliminate the spores through treating your equipment, or you can treat using fumagillin, which is on the market. And we'll get into that on the next slide. So here you can use thermal decontamination, which you want to get your equipment up to 120 degrees and hold it at that temperature for 24 hours. This will kill the nosema spores. So, and then irradiation, it has been known to kill AFB spores, which in turn will most likely kill the nosema spores. And then fumigation with acetic acid, ethylene oxide, and phostoxin. Okay, so there's only one treatment available to treat nosema on honeybees in the United States, and that is up until fumagillin. It is the only approved treatment for nosema in honeybees. It was removed from the market last year, but it's now available again as Fumidil V. And although it was removed from the market, then brought back, beekeepers never reported a huge colony loss due to nosema in that year where fumagillin was not available. Okay, so on this graph, you can see the spore loads in millions on the y-axis and the years on the x-axis. So what stands out here is seasonal load swings occur regardless of treatment, regardless whether the beekeepers treat it or not. And there's no significant difference between treated and non-treated. The graphs are fairly similar. I mean, you can see the peaks and the declines, they're very similar. So in conclusion, with nosema, it's all a matter of preference for the beekeeper. Like, do you treat or do you not treat? That's the real question with nosema. Do you want to treat or not? And I think our next spokesman is Dan Wines.
Yeah. Good afternoon, good morning. Dan Wines here. I am the tech team field specialist based in Michigan, kind of covering the Great Lakes region. And I'm going to talk about tracheal mites. Here we have this this photo just to kind of see this. So this kind of stripy appearing tube is an extracted trachea from an adult honeybee, and these kind of sort of roughly oval shape, football shape, kind of darker patches within that trachea. Those are the actual mites. So we get a sense we're dealing with something pretty small here. It's unlike the varroa mite. It's not something we're going to pick up with our own eyes. It does require a microscope, and we'll talk about that in a minute. Just to give a bit of brief background on this parasite, it was first discovered on the Isle of Wight, which is just an island off the Great Britain. 1921, they'd been having through the 1900s, 19-teens, they'd been having a lot of high losses. It kind of really spawned a lot of advances in bee science and investigation, trying to determine what these high losses were caused by. And in this investigation, they were dissecting bees, and they found these tracheal mites within honeybees. And so that was where they were first described. It was kind of later determined this wasn't the cause of these high losses, but it did lead to the discovery of this parasite. Interestingly, shortly after they were discovered, the United States passed the Honeybee Act of 1922, which banned the import of honeybees from infected countries. So they kind of an early on the biosecurity measure there. It did spread through Europe pretty quickly after it was found, or it was found in Europe shortly after it was found in the United Kingdom. Despite kind of these bee shipment regulations in the United States, it did cross the Atlantic, was found in Brazil in the 70s, and then in Mexico in 1980. First confirmed finding in the United States was in Texas in 1984 in the summertime, and by that fall, it had been found in a number of different states. So it was pretty common with the ways bees get moved around the country for pollination, for honey production, for wintering locations, for shipping of nukes, packages, queens, things like this. Once something kind of gets here, it's everywhere pretty quickly. So this next slide kind of shows us what happened. So this is from the USDA Bee Diagnostic Lab, and what it shows, these blue dots represent essentially each year the percentage of colonies that were the percentage of samples that were infested with these tracheal mites. So we see this black line here, 1984, when they first arrived in the United States. It's very low, it's pretty rare, but immediately the next year we're up in the 35% range, and that number stays pretty steady in the 35-40% of samples analyzed had tracheal mites for about a decade. We go from kind of the mid 80s into the mid 90s. Now the second line I put on this graph, this vertical red line, is 1987. This is when the varroa mites turned up in the United States, and similar story, they were kind of detected in one place, and then within a matter of a couple of years they were pretty well everywhere. As we went through the 90s, it became increasingly imperative that beekeepers treated for varroa mites to keep their colonies alive and healthy. And what the thought, why we have this, as we kind of see this, as we look at the blue dots, this kind of essentially a trend line down to zero, is that in being forced to treat for the varroa mites, beekeepers were actually killing the tracheal mites as well. Kind of, if you're familiar with the concept of like bycatch, you're targeting one thing, but also in this case happens to be a positive outcome is we're killing this tracheal mite as well when we're using treatments to kill varroa mites. So we see this essentially trend line to zero in the within the last decade. They're rare enough that the National Honeybee Survey, APHIS, removed them from the monitoring efforts in 2011 because in 2009 and 2010 they were not detected in any sample within that survey. And currently the USDA will still process samples to look for them, but they report about a 1% positive finding of tracheal mites on an annual basis. So not something that's particularly common these days, but it was, to say, a couple decades back, it was a major part of beekeeping. So just a little bit on this, it's a parasite. On our next slide here, I have kind of a close-up picture of it. And it does live exclusively, as far as we know, exclusively on Apis mellifera, or European or Western honeybees. It's an internal parasite. It lives exclusively in the trachea of adult bees, except for there's a brief period where they transfer bee to bee, but they're living in the bee in the tracheal system, which is part of the respiratory system, and in that trachea system, they essentially are chewing a hole through that trachea to access hemolymph, which is what they feed on. This next slide kind of outlines the life cycle. So it basically begins when a female mite detects. She's able to detect air, kind of the outward breath of the bee, if you will, from a spiracle, which is kind of a hole at the end of a trachea in the bee's thorax. So it's kind of the end of the line in the respiratory system. And so our female mite is able to detect that, and she's able to detect that she's able to pick out a young bee, typically between like 1 and 4 days old, that she's going to enter the respiratory system of. Once she's in there and lodged in the trachea, she lays eggs. Typically, it's about half a dozen within the first few, 3 or 4 days. She'll lay both female and male mites. Typically, the female mites outnumber the male mites 3 or 4 to 1. The males do mature a little quicker, 11 to 12 days. Females are 14 or 15. But once they're both male and female mature, they do mate, and that kind of process takes about 3 weeks total to get them eggs laid, matured, mated. They develop through from egg to larva, nymph, and then finally adults. They mate, and those mated females exit the bee. This bee now started as a young bee a few days old when it was infested. Now it's somewhere in the range of approaching a month old, so it's kind of an older, maybe it's a forager now. But this mature mite exits the bee. She will kind of make her way to the end of one of the hairs on that bee's thorax, and she will look for a new young bee to reinfest and start that cycle over. And because of this kind of old bee to young bee transmission, it was kind of determined that most of this bee to bee spread within the colony occurs at night, or when there's inclement weather, rainy days and things like that, or cold temperatures when the bees aren't flying. So, and if we think about this, this makes sense, because this is a time when we have that high concentration of bees. Kind of everybody's at home if nobody's flying, and we have that contact between the old bees and the young bees all being kind of tight together in there. So it's easier for that mite to be riding on a hair of an older bee and detect and then jump onto a younger bee. So it's kind of the basic cycle of the way this mite kind of progresses in its life cycle and transmission from bee to bee. As far as its impacts on the adult bees, it's one of those, it's tricky because there is no reliable visual indicator. Sometimes you hear reported K-wing, which came up earlier. This is kind of where the bees have 2 pairs of wings, and you'll see just in their resting position, like this bee on the right, its 2 sets of wings are splayed apart. Normally they have between their wings, there's a little piece of anatomy called a frenulum, which kind of it hooks the hind wing and the forewing together, so they kind of appear as one set. But when these are splayed out like that, that can be a sign of tracheal mites. Another potential visual indicator is a distended or kind of bloated abdomen. Sometimes tracheal mites can lead to crawling bees at the entrance. But as we've kind of discussed in these other stressors of the colony, these are not reliable and specific to this particular, they're not specific to tracheal mites. There's a lot of things that can cause bees to present these visual symptoms. As far as colony level impacts, beekeepers report, there's kind of this thought of like it's the colony's failure to thrive. Everything is there that should be there for healthy growth. It's a time of year when you'd expect your colony to be, building and growing. There's good food in the environment. The weather's favorable. It's a time of year when you would expect a lot of growth. You don't have, varroa mite issues or other things like that. And the bees are just kind of stuck and stalled, and they don't go forward. You see diminished brood area, the population's smaller than it should be. They don't grow. The honey yields are not going to be there, even if it's there in the environment for them, and they're just not able to go out and gather it and put on that surplus weight. And then particularly problematic when we get to the winter, we see a looser cluster. It's so our winter bees need to cluster together, and they're in doing so, they're thermoregulating to essentially survive the winter in cold locations. And with this ability to thermoregulate being disrupted, it disrupts that clustering behavior and leads to an increased winter mortality, particularly in northern locations. When the bees, or when the tracheal mites first arrived in the States, it was really much more problematic for beekeepers that overwintered in the northern regions. They saw a big spike in losses due to this kind of disruption of clustering behavior. So again, a lot of things in colonies can kind of lead to these, a lot of stressors will lead to diminished brood area and, colonies not growing. So again, no real reliable visual diagnostics, but these are kind of some of the things that you may be looking for that may, put in your mind tracheal mites. What is required for detection or for diagnosis is dissection. Here we see this photo on the left is a slice of a bee's thorax, and these kind of two roughly vertical white tubes that kind of have a little bit of a V kind of kink in them. Those are the bee's trachea part, which make up the respiratory system there. And we can see on the right, these are kind of segments from the trachea that have been removed from that thorax. And the top photo on the right, those little kind of discolorations, the kind of yellow, darkish kind of lumps, those are actually the mites in there. So when the dissection is looking for mites, they're looking for discoloration, cloudiness, kind of scarring in the trachea. Normally they should be kind of clear and uniform in color with kind of no abnormalities. And then this picture at the bottom is just, again, another piece of trachea that's been removed with those two little, blips on the bottom right there outside the tube. Those are just to kind of give a sense. Those are tracheal mites. It gives you a sense it's fairly large relative to the trachea that it is infesting. So that's what's required to kind of get a confirming diagnosis. When I say like the USDA reports, 1% detection rate, they're doing this kind of dissection to look for the tracheal mites. As far as prevention, they do spread between colonies via drift. As I say, that bee to bee interaction is how they spread from individual bees, and that's how they're going to get from colony to colony. So just thinking about laying out your apiaries in a way to minimize drift. Also not inciting any robbing events where you're getting a lot of interaction and transfer from bees between colonies. Those kind of just general good beekeeping practices for a lot of different reasons, but it can help there. There's also been, some genetic help through bee breeding, where they've kind of found that bees that practice grooming behavior have a better resistance to tracheal mites. And that makes sense when we think about this little brief window where the mite is outside the bee on that hair, looking to get from its old bee to its new host of the young bee. So bees that are grooming are going to break that transmission. And really, though, the main thing, as we kind of saw in that graph that I opened with, it is the Varroa control that has really brought the tracheal mites down to kind of a rarity these days. So good management practices with Varroa, being on top of it, using multiple methods, and being aggressive on controlling Varroa seems to be in that kind of bycatch. By doing that, you're very likely that you will kind of prevent tracheal mite issues as well. Having said that, there is one treatment approved for tracheal mites. As with anything, just like with our Varroa products, it is important, the label is the law. It's important that we understand how and when to apply these, both for bee health, but also for human health, for our health as beekeepers, but also not to contaminate honey or things like that. So the treatment that is labeled for tracheal mites is menthol crystals. They're applied in a kind of a mesh bag. They're available from bee suppliers. Just as we think about with our Varroa mite treatments, there are some that can be used with Supers-On and some that cannot be. This one for tracheal mites is something you cannot use with Supers-On, so it's going to be an early spring or late summer treatment. Typically the treatment is between 15 days and 6 weeks. Again, this is where you want to read the label and make sure you're applying it appropriately. Like we see with some of our Varroa treatments, there are some temperature considerations. So this one, the recommendation is that between daytime high temperatures of 60 and 80 degrees, the bag of menthol crystals should be placed on the top bars of the top brood chamber. There is also a note that above temperatures, if temperatures are anticipated to exceed 80 degrees, that instead this bag of menthol crystals should be placed at the bottom of the hive. So, and we do have one, if you get a positive diagnosis for tracheal mites, there is one product out there that is approved to treat them. So that's kind of the rundown of this pest. And, you know, just to kind of summarize before we move to questions, what we've run through here, and say tracheal mites, they were an issue. I think they're an interesting kind of part of you know, the beekeeping story of the last several decades, but they're not something we really need to be too concerned about today. I think it's good to be aware of them, but they're not a major stressor at this point. As Natalie talked about with the colony collapse disorder, it was a big deal, you know, 14, 15 years ago. It's not something that, you know, we really are currently seeing. That, you know, we get a lot of reports, but as she said, a lot of this is kind of the misconception and co-option of this term and what it actually means, and misuse of this term in the media. So it's good to be aware of what it was, and it's not to say that we may not see these things come up again, but currently it's not something that's a major concern. Nosema is a little more of a middle ground, as Cade kind of talked about. There are some impacts on colony health, but that you know, there's a lot of unknowns. It's one that we as beekeepers and bee scientists kind of puzzle and scratch our head on. You know, what the Nosema numbers mean, and should we be treating, and if we are treating, are we getting good value out of that treatment? Are we achieving our objective? So that's one. There's plenty of ongoing work to understand this Nosema. I say when we transferred or switched from kind of Nosema apis to Nosema ceranae, it was kind of caused this. We need to relearn what this parasite is and how it impacts our colonies and what we as beekeepers can do. And then the last one is, as Rob talked about, the viruses. These ones are what I see as a beekeeper and working with beekeepers and bee scientists. These viruses they're big problems. You know, they are very much in the category of we're learning more every day about them. There's a lot of work going into understanding them because they are so problematic. It's really become a research priority of a lot of scientists because, you know, say we're starting to understand these mortality more, and it's not just the varroa mites, it's the viruses that come along with them, and the long time that it can take a colony to kind of rebound once they've been seriously impacted from this. So that's kind of the overview of what we talked through. I think we've got about 15 minutes left here for questions, so we can open it up to those, I think.
Thank you so much, field specialist. Thank you to our sponsors and collaborator. We will take some questions. What I did is I kind of looked at questions as they were coming in and tried to pool them with the different categories of the different parts of the talk that we had today. Before we get to questions, I want to thank you all for joining us at the webinar this morning. I don't know if you noticed, but a couple of our field specialists are actually in the field. And so they stopped their sampling with the colonies to just kind of join in on the Zoom here and give you some information about these diseases. So a big shout out to our field specialist. I always feel like I work with the crop, the cream of the crop. So I'm thanking them for their time and all of their knowledge, passing that on to us. So I'm sure that you're all applauding really loudly here. And we're gonna move to some questions. I don't know if we're gonna have time to talk about them all, but if there are some follow-up questions, please email us or let us know how else we can help you. You will also notice on this slide here that we have a please consider supporting the Bee Informed Partnership and the donate button. It's not very bright, but you can go and find that donate button on our website as well as on our Facebook page. Make sure that if you think that you see value, if you do see value in these presentation, we're a small nonprofit organization almost relying solely on grants and donations, so please help us out. All right, moving on to questions. So I'm going to start with a couple. We're going to go back to the beginning when we talked about CCD. And so Natalie, I have a few questions for you. So one of the questions which I thought was interesting is what is driving the increase in colony numbers worldwide? So can you go back and maybe just discuss a little bit what you feel colony numbers worldwide are looking like?
Yes, so this is data that has been collected by the FAO, Food and Agriculture Organization worldwide. So they do collect information from every country that might be collecting those numbers differently from one country to the next. That is the best collated information that we have. Actually, a lot of those increase in colony size are due to specific countries. We believe that it's because of the demand and in pollination. So yeah, you can actually go on the FAO website and tease out that information and look at country by country, region by region, which are the area in the world that are driving this number up, if that's something that interests you. We believe that is, as I said, probably due to the need from pollination, and just, yeah, as I said, it might be hard to compare from one country to the next, but it is one of the best estimates we have. One thing also that we want to point out is that the number of honeybee colonies doesn't mean that all of those honeybee colonies are thriving and healthy. So as all estimates, just take it with a grain of salt. The main message in that graph was that honeybees are not going extinct. They are a global species present everywhere in the world. And so we know that there is no risk at population decline and extinction, even despite the mortality rates that we are seeing.
Thank you, Natalie. There was also a couple of questions that I'm going to try to combine. Some people are asking if you can see all the signs of CCD, but like perhaps some of these infections that led them to leave were mite-borne. So some of the participants say that they've seen all these signs of CCD, but they also suspected parasitic mite syndrome or another disease that may have made them leave. So what do you think about that?
So yeah, that is tricky. That is why the last symptom, which is no varroa mite infestation, was added to the case definition, because at the time where the case were defined, they ruled out varroa so they knew that it wasn't because of varroa from those case definition. Now, as we have said, throughout the work that our field specialists are doing, they have been called to, I guess, scenes of the crimes where they were asked to see if it was CCD, and that was their experience, I believe, and maybe they can chime in with their own observation, that they have a lot of time they have seen cases where colonies collapsed very fast, so it looks like colony collapse disorder, but they were able to attribute it to past varroa infestation that probably transmitted a lot of viruses, and then those, because the population crashed, the mites themselves were not seen, but you can still see in your colonies, if you used to have high varroa infestation, there are other signs that you can see in your broods. Like, for example, you might see some varroa frass still present on the frames. If you want to know what that looks like, you can look at our varroa webinar. We have some great pictures of those. So even though those bees and mites were not present anymore, there are still signs of leftover that you can identify past high varroa infestation, if that is something you want to rule out.
Thank you, Natalie. All right, Rob, I have a question for you. Somebody asked if they have equipment from a hive that did die from viruses. What could you do with the equipment? Could you freeze it, heat it, both, or not necessarily do anything about it?
You know, the beekeepers out here, they tend to just try to use the equipment right away. I don't for a fact know if viruses are transmitted through equipment. I would think there is a means that could happen, but most likely if you put the equipment on something that's strong, they're going to clean it all up before she uses it. And I haven't really seen where somebody uses old equipment and you see viruses pop up, but we're not always taking viral samples. But I'm just thinking of visual viral symptoms. I've never seen a beekeeper put on old equipment and see virus symptoms pop up. And I haven't gotten any reports, and we work with a lot of large-scale beekeepers, and if there were something to that, I think they would let me know.
All right, that probably also answers the question that Janine sent about deformed wing virus and how long does the virus persist on comb and honey and pollen. I don't think we have data on that, correct?
That would probably be a Natalie question, but again, you know, usually the virus is you're seeing this stuff in the bees. I don't know how much they're picking up from old comb. You could freeze it, but I don't know if that's necessary. It's almost better if you have dead out equipment to stick it on another colony right away than to store it at all. From what I've seen, and maybe Natalie, you have some information.
Yeah, we can add to that it is not a bad practice to freeze your equipment before reusing if they've been away from bees. Definitely for bacterial disease, and that is just part of good management practices, because we definitely know that bacterial disease get transmitted by old equipment. So if you do that for bacterial diseases, you might that might be part of your practices generally. In terms of viruses, there is no evidence that it transmits from equipment, but as I said, because it happens for bacterial disease, it should be part of your good practices anyway.
I will say, if you see moldy equipment, they will clean that up, but I have seen them get sick from that, and I've seen some chalk brood pop up, and we kind of thought that it was because the frames were really moldy and didn't look so good, and we think that kind of weakened the bees a little bit, and we saw a little bit of chalk brood, but that was the only case that I've seen in the past several years where I felt like that old equipment was causing an issue to the colony.
All right, we're going to move to a few Nosema questions. Carolyn asked, what is the safest way to clean off the dysentery from a hive, or should we just let the bees handle it?
Okay, I would say you can just use soap and scrub it off, or I mean, sometimes people just leave it. So either one of those options. If you really want to clean it, I would say just scrub it off with some soap.
Yeah, and if you add a drop of bleach, the bees are go and clean it all up afterwards too. So maybe be careful about what you use in that cleaner. I thought there's another question from Holly that she asked. She thought that you needed a really super powerful microscope to see some of these Nosema spores. Does anybody know what kind of magnification you need on a microscope in order to see Nosema spores?
I believe it was only 400 times magnification, and you can see the spores. But you do have to be careful, because you are crushing up the insides of bees. You have to be careful not to misinterpret what you're seeing between Nosema spores and pollen grains, because they can look similar.
Yeah, and sometimes just little air bubbles can also look a little similar. So anybody else on the team want to comment on that? And do we need a stain? I didn't see if we needed a stain.
No, you don't need a stain to see it. Your best option for Nosema is if you know of a beekeeper in the club or have a sample that has Nosema. That's the best way to learn how to identify the spores, is to have a known sample that has it and then compare the two. But that's the best recommendation I have for being able to identify it. It is kind of hard to identify it first, but once you know what the spores look like, it's fairly simple. And there's quite a bit of literature online that can help you identify them.
Sounds great. Thank you. Rob, can you also talk, because I know you mentioned that yesterday in some of our discussions, will UV irradiation kill Nosema spores?
I would assume so, because it kills, the American foul brood spores. But I do know FOS toxin that is supposed to be active against Nosema. But I think irradiation would probably be a good method to use on old stored comb if you have means to use it. It's not available everywhere, but it probably is a good practice, I would say.
Somebody asked if Nosema can be transmitted to human. I don't know that answer. Does anybody know that?
I think it's safe to say that it is not the case. There are some questions about transmission to other non-honey bees, so other non-apis bees. But actually, there has been recently a lot of work in actually just renaming the whole Nosema genera, and it might not even be Nosema anymore. It might be completely renamed and associated with a different part of the phylogeny. So. So no on honeybees, no on honeybees to human transmission. That is.
Okay. Somebody, and I believe that we may have mentioned this real quick, but is fumagillin effective against Nosema ceranae or only Nosema apis? And I know, Natalie, you had read some information on that, so maybe you can illuminate.
Yes,
this.
so the problem is a lot of the research on Nosema, as Kate mentioned, is that a lot of our old information, like most of the literature information came from studies done on Nosema apis. And since we have now switched, and Nosema ceranae is the most prevalent, if only, species in the United States, a lot of that old literature is now irrelevant. So fumagillin was developed for Nosema apis. It was proven to be effective against that, and there is only conflicting information now on Nosema ceranae. So some studies say it is effective, some other studies contradict that. So I guess the jury is still out. And as I said, our own data, we don't see a lot of difference in resulting loads between beekeepers that use and don't use treatment. So we don't want to discourage it if people consider that they want to use it because they feel it puts their colonies at risk and they want to treat. We are on the fence until we can see better research on that topic.
And Natalie, can you also confirm, so somebody wants to know what happened to Nosema apis. Why is it that it's not so prevalent anymore and that Nosema ceranae is the one we see?
Yes, absolutely. It is a classical example of displacement of species. So Nosema ceranae outcompeted Nosema apis and replaced it. So actually, there has been studies in Canada where they still have Nosema apis. So it might be that Nosema ceranae arrived in the United States, took over in our region and pushed Nosema apis north. And I know that they still have Nosema apis in Canada, but we don't have it anymore in the United States. So it was outcompeted.
And can everybody confirm, I do believe we do not have any data on how Nosevit as a treatment for Nosema works, but can everybody confirm that for me?
Yeah, I'm not aware of anything that definitively shows one way or another that it impacts Nosema ceranae.
All right, we're going to move to you, Dan, with a couple of tracheal mite questions before we finish today. Reed wants to know, how do young tracheal mites leave bees?
Young tracheal, okay, so well, they're leaving once they're mature after they've completed that kind of 3-week cycle roughly. They're leaving this the same way. They're going out through the spiracle, and which puts them out on the thorax, and then they're climbing to the hair on one of the hairs on the thorax. So same point of exit as entry, as I understand it.
Okay, and Karen wants to know which of the Varroa treatments shows the most deterrent effects on tracheal mites.
So I don't really specifically want to comment on those. I would say if you're concerned about tracheal mites and do have them, you know, and you want to target that pest, menthol is going to be a good option. There are reports of a variety of Varroa miticides being effective, but it's really going to be practicing. You know, your best practice with Varroa mite management should include using multiple different chemicals with different modes of action. So that may be, you know, formic in the summertime and something like, you know, apivar in the fall and oxalic. So by kind of mixing and coming at them from different angles, these are, you know, seem to have been effective, because this is kind of what's taken place out of necessity in the industry. And that seems to have gotten us to where we are today. So I don't know that there's specific, you know, say a kind of bycatch concept, but a broad, varied approach to treating varroa mites seems to be doing a good job at catching tracheal mites as well.
And Christy wants to know, what magnification on the microscope would you need to see tracheal mites? I
believe some of the photos I showed were taken at 40x, but there was some that were also labeled at 10x. So not nearly as much as with the Nosema. They're you know, kind of at least an order of magnitude bigger.
Okay, and Anthony wants to know if it is still suggested to use grease sugar patties for treatment against tracheal mites, or is it more of a cultural control measure?
So that's something, you know, that's kind of, you do come across that, and beekeepers say they've done that, but it's not something, you know, we can't just go applying any, you know, random thing out of the kitchen pantry to our hives as a, you know, with the intent of controlling a pest. You know, their pesticides have labels for a reason. So I would say I can't recommend, you know, something that's going to be a off-label treatment for this pest when there is an on-label way to do it.
Okay, thank you. We have a couple of questions that are related to, I think, all of them. And so I'm going to put the question out there. And so what is the minimum recommended colony inspection frequency to keep all these issues at bay?
So it is kind of realizing the answer, like all things with bees, is it depends. And this one, it kind of depends mostly on time of year and your location. I mean, I kind of think of it as there's some critical periods in the spring where I'm probably in them every 10 days or so. And then there's periods in the summer once they're off and running, and then in, you know, honey production mode every 3-4 weeks is fine. There's kind of this get them off on the right foot and stay out of their way. And then it gets real critical again in the late summer into the fall, a little more frequency, because you want to be monitoring, particularly the varroa mite levels. They can change pretty quickly that time of year, and it's real critical. So, you know, I think if you wanted one number, I'd say 2 to 3 weeks, which I realize is a range, not a number, but somewhere in that vicinity. And then, of course, you know, winter, if you have a winter and your bees are dormant, like where I'm at in Michigan, I don't touch them from, you know, November through maybe March. When there's a nice day, I'll pop a lid and see how they're doing on food, but I'm not really poking in them. So very seasonally dependent.
And do any of these diseases affect the honey usability?
Not that I'm aware of. Where you would potentially be impacting your honey is in your application of some of these treatments or remedies. If you weren't following the label, there's potential for contamination. So that's you know, whether that's fumagillin, you know, that is an antibiotic. That's not something we want to end up in honey. So we're not going to be feeding with Suprazon, or for a period prior. I forget the exact withholding period, but it's several weeks, I believe. Same thing with like the menthol crystals for tracheal mites. There's the potential to contaminate the honey. And again, with all our varroa miticides that we might use to, say, keep our viruses in check, there are potential contamination issues for a lot of them, but it's not the actual stressor itself. It's more our reaction as beekeepers that has the potential to contaminate that honey.
And I've had this question from a couple of participants. Can you use essential oils for some of these diseases? Probably most likely for Nosema. It was asked for Nosema, but it was also asked for tracheal mites and even varroa. So what do you think about using essential oils for treating some of these diseases?
It's another one where it's not labeled for treating these kind of, you know, pests, parasites. So it's not legal, but also I'm not aware of any kind of scientifically backed findings that they are effective in doing so.
All right, we're gonna wrap things up here. There is one more question if you guys want to stay, which is related to something else, but I do feel like I need to make a little distinctions. I've had quite a few questions relating to EFB. EFB is a brood disease, not an adult disease, and we did have a webinar a few weeks back on identification of brood disease, during which we had an entire section solely on EFB. So if you are interested in learning a little bit more about EFB, I suggest that you go and review or view our recording of the brood disease identification webinar that we gave a few weeks back. You can find this on our website at beinformed.org/educate and under the BIP Talks, or you can also find it on our YouTube channel, the Be Informed Partnership YouTube channel. One more question for you, Natalie. Anne Fraser asks, what does BIP think is the cause of the higher reported summer mortality in 2019? And that is right up your alley. So I thought I would just finish up with this question.
Yes, so Fraser is mentioning the latest result in our survey, which came out from the survey that we ran this April. So this past year, which is 2019-2020, was quite unusual because we had a low winter mortality, but we did had a high summer mortality last summer. And so we think it's important to keep doing the survey every year because it's really important to keep a pulse on those rates to see if they're going in one direction or another. And we can say that since we've been running this survey for like 12 to 13 years now, we do not see that it's getting worse or it's getting better. So there is a cycle that we have some years that are better, some years that are worse. So there is some fluctuation in loss rates, and that is what we saw last year, in that the summer was particularly high last year. We do have some hypotheses as to why that might have been, but this is also where the speculation enters. So we have a hard number, but we wondering why it came about now enters our hypothesis, right? So one of the things we have noticed is that the winter before, so the 2018-19 winter was a very bad winter. So this might have been the leftover from that winter. We also know that that spring, 2019, was a really wet and cold spring, and that generally we had observed in the beekeepers that were working with us that the colonies were slow to pick up, and they didn't really build as strongly and as quickly as they could have. And so that is usually the time period of when colonies will pick up and start and really outrun varroa, and they didn't really have that chance that year. A lot of queen breeders had a very particularly bad year that year. And so a lot of beekeepers were waiting for those queens, and so that contributed to just a delay in the pickup of the buildup of those colonies last year. One of the tech team wants to add to that. Or not? Yeah, generally that's just that is where we were going, trying to explain those numbers that we observed. On a nice note, as I said, this is a cycle. We know that there are worse and better years, and I say better with being careful, because that still means 30% loss, which for a lot of beekeepers is unacceptable. I guess this is part of the new normal where we started to because we see those high level of losses every year, it's becoming more usual to see them. We understand that means that there is still a lot of stress on the beekeepers to sustain those high levels of losses year after year. And so, yeah, I guess it's
excellent. Well, thank you, Natalie. Thank you, everyone, for participating today. I think that we are going to wrap things up. We're already a little late, but I do see that many of you stayed on. So my guess is, you found it useful. So thank you again for your participation. We will have the recording for this probably on our website by tomorrow or on the YouTube video. So, and if you still have some questions that we were not able to answer throughout our question and answer section, please feel free to email them to me, Anne-Marie Fauvel at the Be Informed Partnership. You can also find my contact information on the website. So thank you again. Have a wonderful day. And we look forward to doing more of these.